Health authorities and virologists are issuing a renewed warning this season regarding a low-frequency but high-consequence pathogen that often hides in the most mundane of domestic settings. While many associate infectious disease risks with international travel or crowded urban centers, one of the most significant threats currently monitored by agencies like the Centers for Disease Control and the Pan American Health Organization originates in the quiet corners of garages, sheds, and attics. The narrative of hantavirus pulmonary syndrome, or HPS, often begins with a common weekend chore, cleaning out a storage space that has remained undisturbed for months or years. For many, the first sign of trouble is not a cough or sudden respiratory distress, but rather a profound set of muscle aches and fatigue that many victims initially mistake for the physical exertion of their cleaning tasks. However, public health reporting indicates that by the time these symptoms manifest, the window for effective medical intervention is already beginning to close. This is why medical experts emphasize that muscle aches following weekend cleaning could mean a patient has only moments, or perhaps hours, to seek the level of intensive care required to survive a virus with a case fatality rate that can exceed 40% in certain regions. In 2025, the Americas region reported an aggregated total of 229 cases and 59 deaths, maintaining a regional case fatality rate of nearly 26%. The geography of the threat is broad, with confirmed cases recently spanning Argentina, Brazil, Bolivia, Chile, Panama, Paraguay, Uruguay, and the United States. To understand the lead and framing of this public health issue, one must look at the specific mechanism of transmission, which is fundamentally tied to rodent ecology. Hantaviruses are not new, but their impact on human populations remains severe because they are zoonotic, meaning they jump from animals to humans. The primary reservoirs are specific species of rodents, such as the deer mouse in North America or the long-tailed pygmy rice rat in South America. These animals shed the virus in their urine, droppings, and saliva. The critical danger for humans occurs when these waste products are disturbed, often by a broom or a vacuum during a seasonal cleaning, causing the viral particles to become aerosolized. Once airborne, these microscopic particles are easily inhaled into the lungs. From that point, the virus begins a silent incubation period that can last anywhere from 1 to 8 weeks, though 2 to 4 weeks is the most common time frame. During this period, the patient feels entirely healthy, unaware that the virus is replicating within the endothelial cells that line their blood vessels. The framing of this crisis by the World Health Organization and local health departments focuses on the transition from the prodromal phase, the initial flu-like stage, to the cardiopulmonary phase, where the virus triggers a massive inflammatory response. This response causes the tiny blood vessels in the lungs to leak fluid, effectively causing the patient to drown from within. Because the early symptoms of fever, headache, and myalgia are so nonspecific, they are frequently misdiagnosed as common influenza or even COVID-19, leading to catastrophic delays in treatment. As we move into the core developments of 2026, international surveillance has shifted toward a specific and highly concerning cluster of cases that has challenged previous assumptions about hantavirus transmission. On May 2, 2026, the World Health Organization was notified of a cluster of severe respiratory illnesses aboard a cruise ship in the Atlantic, which had departed from Ushuaia, Argentina. As of May 15, this outbreak has resulted in 9 confirmed cases and 3 deaths. While most hantaviruses, including the sin nombre strain common in North America, do not spread between people, laboratory sequencing has confirmed that this particular outbreak involves the Andes virus strain. This development is significant because the Andes strain is the only known hantavirus capable of limited person-to-person transmission, particularly among those in close, prolonged contact. This has triggered a massive multi-country response involving the repatriation of passengers to countries like Spain and Switzerland, and medical evacuations to clinics in Johannesburg, South Africa. In Spain, health officials reported a passenger who tested positive via polymerase chain reaction, or PCR, and was immediately isolated. This international sprawl highlights the logistical complexity of managing a hantavirus outbreak. as samples must be handled under maximum biological containment conditions and transported using a strict triple packaging system to prevent accidental exposure to lab personnel. The data provided by the Pan American Health Organization for the late 2025 and early 2026 period shows a disturbing trend in lethality. In Brazil, the case fatality rate has reached 32%, which significantly exceeds the 4-year average of 15%. Similarly, in Paraguay, the lethality rate rose to over 22%, with an outbreak in the Mariscal Estigarribia region linked specifically to road construction activities that disturbed rodent habitats. These figures represent more than just statistics. They reflect a strain on regional healthcare systems where early diagnosis remains elusive. In the United States, although the total number of cases remains low, averaging about 3 per year in states like California, the mortality rate remains high, with nearly 4 out of 10 infected individuals succumbing to the disease. The contrast between claims of a routine flu season and the verified clinical data is stark. In the cruise ship cluster, the case fatality ratio was calculated at 38%. Clinical reports from these cases describe a rapid deterioration. One adult female passenger disembarked in St. Helena with what appeared to be minor gastrointestinal issues, only to die 2 days later in a Johannesburg clinic after her condition spiraled during a flight. This speed of progression is a hallmark of Hantavirus pulmonary syndrome, where the transition from feeling under the weather to full respiratory failure can occur in less than 24 hours. Surveillance agencies are now closely monitoring these spillover events, where human activity, whether it be leisure travel, on a cruise ship or industrial work in rural departments, intersects with the environmental presence of the virus. Given the absence of a licensed vaccine or specific antiviral treatment that can cure hantavirus pulmonary syndrome, public health guidance focuses almost exclusively on rigorous prevention and early clinical intervention. The limits of current medical knowledge are honest and clear. While ribavirin has shown some efficacy in treating hemorrhagic fever with renal syndrome, a different manifestation of hantavirus found primarily in Europe and Asia, it has not proven effective for the pulmonary form prevalent in the Americas. Therefore, the primary strategy recommended by the CDC and the World Health Organization is the management of symptoms through intensive care. This includes the use of supplemental oxygen, intubation, and, most critically, extracorporeal membrane oxygenation, or ECMO. Data indicates that when ECMO is initiated at the earliest sign of heart or lung failure, the survival rate can reach 80%, even in patients who have experienced a complete cardiopulmonary collapse. However, this level of care is only available at advanced medical facilities, making early referral and the communication of potential rodent exposure to doctors a matter of life and death. If a patient waits until they are in full respiratory distress, the likelihood that even the most advanced treatment will be effective drops significantly. Prevention, therefore, remains the most powerful tool in the public health arsenal. For those undertaking weekend cleaning projects in garages or sheds, the guidance is specific and must be followed to the letter to avoid aerosolizing the virus. Experts advise against the use of vacuums or brooms in areas where rodent activity is suspected. Instead, the area should be thoroughly ventilated by opening doors and windows for at least 30 minutes before entering. Protective gears, specifically an N95 respirator and rubber or plastic gloves, is considered essential. The recommended cleaning protocol involves soaking any droppings or nesting materials in a disinfectant solution, typically a mixture of bleach and water, for at least 5 minutes before wiping the area with a damp paper towel. This wet cleaning method ensures that viral particles remain trapped in the liquid rather than becoming airborne. All waste should then be sealed in plastic bags and disposed of immediately. Beyond cleaning, officials urge the sealing of any gaps in homes larger than a 1/4 inch to prevent rodent entry, and the storage of all food in rodent-proof containers. As we move further into 2026, the message from health agencies remains one of calm but heightened awareness. While the risk to the general public remains extremely low, the high fatality rate for those who are infected means that domestic hygiene is no longer just a matter of aesthetics, but a critical component of infectious disease defense. By understanding the link between that weekend cleaning task and the sudden onset of deep muscle aches, individuals can provide their doctors with the information needed to trigger life-saving interventions before it is too late. If you found this report on global public health surveillance informative, consider subscribing for more in-depth news analysis. The clinical progression of hantavirus pulmonary syndrome, or HPS, represents one of the most aggressive challenges in modern diagnostic medicine, primarily due to the deceptive nature of its initial presentation. In the days following the initial exposure, often a high-intensity cleaning event in a garage or shed, the virus undergoes a silent incubation period. According to established medical literature and data from the Centers for Disease Control and Prevention, this phase typically lasts between 1 and 5 weeks. During this window, the patient remains asymptomatic as the virus begins to target the endothelial cells that line the blood vessels, particularly within the lungs. The shift from the silent phase to the symptomatic stage is characterized by what clinicians call the prodromal phase. This is the period referenced by public health officials when they warn that muscle aches should not be dismissed as mere physical fatigue. These aches, specifically in the large muscle groups of the thighs, hips, and back, are accompanied by a high fever and often gastrointestinal distress. For the hospital systems tasked with managing these cases, the primary difficulty lies in the differential diagnosis. In its early hours, HPS is indistinguishable from common seasonal influenza, West Nile virus, or late-stage COVID-19. However, there are specific hematological markers that labs look for to signal a hantavirus infection before respiratory failure begins. These include a significant drop in platelet counts, known as thrombocytopenia, and an increase in the proportion of red blood cells to total blood volume, known as an elevated hematocrit. These signs indicate that the patient's blood is thickening as fluid begins to leak from the circulatory system into the surrounding tissues. The laboratory and testing infrastructure required to confirm a hantavirus case is specialized and highly regulated. Because hantaviruses are categorized as high-consequence pathogens, diagnostic procedures are often split between frontline hospital labs and state or federal reference laboratories. Initial screening frequently utilizes an enzyme-linked immunosorbent assay, or ELISA, to detect the presence of specific antibodies. Specifically, the presence of immunoglobulin M, or IgM, indicates a current or very recent infection, while immunoglobulin G, or IgG, can indicate a past exposure. In more advanced diagnostic settings, healthcare providers rely on polymerase chain reaction, or PCR, testing to detect the viral genetic material itself in the patient's blood or tissue samples. However, time is the critical variable. As noted by the health reporting in the current 2026 cycle, once the prodromal phase ends, the patient enters the cardiopulmonary stage with startling speed. This transition is marked by a sudden onset of shortness of breath and a dry cough. At this juncture, the virus is not actually destroying the lung tissue directly. Rather, the body's own immune response triggers a massive increase in capillary permeability. The microscopic vessels in the lungs become porous, allowing plasma to flood the air sacs, or alveoli. This creates a physiological scenario where the patient is unable to oxygenate their blood, regardless of how much effort they put into breathing. This rapid deterioration is why the window for treatment is described as a matter of moments. By the time a patient reaches the emergency room with severe respiratory distress, their blood pressure may already be dropping toward a state of shock. Surveillance agencies and specialized labs, such as those within the CDC's Viral Special Pathogens branch, monitor the genetic drift and distribution of these viruses to track any changes in their behavior or virulence. While the majority of hantavirus cases in North America are linked to the Sin Nombre strain, officials also keep a close watch on other variants like the Monongahela or New York strains, each associated with different rodent reservoirs. The surveillance effort is not limited to human patients. It involves a complex ecological component where field biologists and mammalogists trap and test rodent populations to determine the prevalence of infection in specific geographic areas. This data allows public health departments to issue targeted warnings when a particular region shows a spike in viral load among local deer mice or cotton rats. The shipping and logistics involved in this research are rigorous, requiring all viral samples to be transported under strict biosafety level 3 or 4 conditions. For clinicians in rural or suburban clinics where these cases often first appear, the instruction remains consistent. Take a detailed exposure history. If a patient presents with sudden onset muscle aches and fever, the single most important question a doctor can ask is whether they have recently disturbed a dusty, rodent-prone area. Without that piece of information, the lab work might be ordered too late, as the transition from flu-like symptoms to full-scale pulmonary edema can happen in as little as 4 to 24 hours. This clinical reality underscores the ongoing uncertainty in hantavirus management. Even with the best modern lab technology, the speed of the virus often outpaces the logistics of the healthcare system. Given the absence of a licensed vaccine or specific antiviral treatment that can cure hantavirus pulmonary syndrome, public health guidance for the 2026 season focuses almost exclusively on a rigorous two-tier strategy: mechanical exclusion of rodents and the strict adherence to wet disinfection protocols. The limits of current medical science are a significant factor in this guidance. Currently, there is no post-exposure prophylaxis available for those who believe they may have inhaled viral particles. Because the virus exists in a stable state within dried rodent waste for several days, any environment that has remained closed and undisturbed serves as a potential reservoir for aerosolization. This is why the garage death trap phenomenon is so persistent. When a homeowner enters a shed or garage and immediately begins sweeping or vacuuming, they are creating a high-concentration cloud of microscopic debris that carries the live virus directly into the lower respiratory tract. To counter this, the CDC and various international health ministries have standardized a cleaning protocol that should be treated with the same level of seriousness as handling hazardous materials. The first practical takeaway for any residential or industrial cleaning task is the 30-minute ventilation rule. Before any work begins, all doors and windows must be open to allow for a cross breeze, which significantly reduces the concentration of any airborne viral particles. During this time, the individual must remain outside the structure. The second critical step involves personal protective equipment. While a standard surgical mask may offer protection against large droplets, it is insufficient for hantavirus. Public health officials recommend a fit-tested N95 or higher respirator, along with rubber or nitrile gloves and long-sleeved clothing that can be immediately laundered in hot water. The most vital instruction, however, is the total prohibition of dry cleaning methods. One must never use a broom, a leaf blower, or a standard vacuum cleaner in these spaces. Instead, the area must be saturated with a disinfectant solution, typically a mixture of 1 part household bleach to 9 parts water. This solution must sit undisturbed on any droppings or nesting materials for at least 5 minutes. This wetting process serves a dual purpose. It chemically deactivates the virus and physically weights the dust particles, preventing them from floating into the breathing zone. Only after this saturation period should the materials be wiped up with paper towels, double-bagged, and disposed of in a secured outdoor trash receptacle on a broader scale. Surveillance and public policy in 2026 have moved toward proactive rodent exclusion as a fundamental pillar of community health. Local health departments are increasingly emphasizing that rodent-proofing a home is not just an issue of property damage, but a critical medical preventative measure. This involves sealing any entry points larger than 1/4 of an inch, roughly the width of a pencil, using steel wool, metal flashing, or caulk, as mice can squeeze through incredibly small apertures. Beyond the home, surveillance teams continue to monitor the ecological factors that drive outbreaks, such as mast ears, where an overabundance of seeds leads to a population explosion of reservoir species like the deer mouse. This environmental data is used to issue early warnings to park rangers, construction crews, and utility workers who are at the highest risk for occupational exposure. The ethical dimension of hantavirus reporting rests on a delicate balance between transparency and public calm. While the lethality of the virus is high, the actual frequency of infection remains low compared to other respiratory illnesses. However, the tragedy of HPS often lies in its preventability and the relative youth of its victims, who are frequently healthy individuals engaged in routine labor. The uncertainty that remains, such as why some individuals develop the cytokine storm that leads to pulmonary edema while others may have milder reactions, continues to drive intensive research in immunology. Until a definitive treatment or vaccine is developed, the burden of safety lies with the individual and their awareness of their surroundings. The ultimate goal of this reporting is not to instill a fear of the outdoors or domestic